since we have talked about an overview of the subphases of the mitosis, so we're gonna look closer of each phases, and we're gonna talk about each event that happens in each phases. So we have in the first subphase is a prophase in prophase. So is the prophase. So the chromosomes are tightly coiled with the sister chromatids joined together, so the nuclearly disappears. So the mitotic spindle begins to form. So it is composed. So the mitotic spindles, it is composed of centrosomes and the microtubules that are extended from them. So the radial array, of a shorter microtubules that extend from the centrosomes are called the asters. This meaning that they're just short microtubules. We have the centrosomes move away from each other, apparently propelled by lengthening microtubules. So meaning that if these two, the centrosomes, they move apart, they cause, the result of cause this microtubules to appear. That's what it does. So here's a picture of another. Here's a picture again of animated prophase or subphase of the prophase. So that's what it is. The second subphase in mitosis is a prometaphase. During prometaphase, the nuclear envelopes during prometaphase, the nuclear envelope fragments or breaks, and microtubules from the spindle interacts with the condensed chromosomes. Now, here's the thing. So we have each of the chromatids of a chromosome has a kinetochore, a specialized protein structure located at the centromere. So the kinetochore microtubules from each post attached to one of two kinetochores. Non-kinetochore again chromatids. Non-kinetochore chromatids interacts with those from opposite ends of the spindle until they are all arranged at the metaphase plate. So an imaginary line plate. So a metaphase plate, which is an imaginary line equidistant from poles, defining metaphase. So that's what a metaphase is. So a microtubules interact with those from opposite ends of the spindle. So the spindle fiber pushes the sister chromatids. So the spindle fibers pushes the sister chromatids until they are all arranged at the metaphase plane, an imaginary plane equidistant from the pole. So, defining metaphase, that's what it is. So here's the metaphase picture. We have the spindle fiber pushes this spindle fibers pushes the sister chromatids together until they're all arranged at the metaphase. They pushes from one side at the North Pole, and the other one, the first spindle, pushes from north. Second one pushes from the south, which brings the sister chromatids together to make, so they pushes together at the metaphase plate, which makes the chromosomes again, or sister chromatids. We have anaphase. At anaphase, the centromeres divide, separating the sister chromatids. Now this is when the centromere divides, separating the sister chromatids. Here's the centromeres now. So now since they are attached, if you go to the last, this is the centromere. So since they are attached in this picture, they move apart, or they no longer are attached together. So each now is pulled towards the pole. Here's one pole to the north again, and to which is attached by spindle fibers, and the other one is pulled again by the same spindle fibers to the south pole. So by the end of the two poles, they have equivalent collection of chromosomes. Now, since they have now satisfied and they have equivalent collection of chromosomes. Now, each one have a daughter chromosomes. This is a chromosome now. They are full chromosomes now. Finally, telophase. This daughter cells at the telophase, the daughter nucleus begins to form at the two poles. So now nuclear envelope arises from fragment of a parent cell's nuclear envelope, and other portion of the endomembrane system. The chromosomes become less tightly coiled. So cytokinesis happens again. So when the chromosome become less tightly, cytokinesis, the division of it, is usually well undergoes by the late. It happens mainly by the late telophase. So where what it does, the cytokinesis, is the cleavage, what it like pinches the two cells and causes each one like to divide spontaneously. So in animals, the cytokinesis involves the formation of cleavage thyro, which pinches the cell. That's what I said. In a plant cell, the vesicle derived from the Golgi apparatus, whatever it is, Golgi apparatus, produces a cell plate at the middle of the cell. It's only so. So in animal cell, they only produce the cell plate at the middle of the cell, and from Golgi apparatus. Here's a picture of telophase and cytokinesis. I mean, this one, it's not telophase 1. It's just a telophase, because we're talking about mitosis, not meiosis. Meiosis is when we have a telophase 1. So I just kind of make a mistake. It's just a telophase and cytokinesis. It just happens again, cleavage or thyro. That's what we talked about now. So we have mitotic spindle. Let's look at closer look. The mitotic spindle in an apparatus of microtubules that controls the chromosomes move during mitosis. During prophase, assembly of microtubules begin in the centrosomes, the microtubules organizing center. So the centrosomes replicate, forming two centrosomes that migrate to opposite ends of the cell. So at the spindle, microtubules grow out from them. An aster, a radial array of a short microtubule, extend from each centrosome. So the spindle includes the centrosomes, the spindle microtubules, and the asters. That's what the spindle includes: the centrosomes and the spindle microtubules and the asters. During prometaphase, some spindle microtubules attach to the kinetochore of chromosomes and begin to move the chromosomes. So at metaphase, the chromosomes are all lined up at the metaphase plate, the midway point between the spindle's two poles. Here's the picture again, the mitotic spindle at the metaphase. Let's talk about, in this diagram now, where we have, the diagram, it shows two duplicated, chromosome array at the metaphase plate. Here's the two duplicated chromosome array, at the metaphase plate. This is a metaphase plate. We were talking about here. This is a plate actually. Two metaphase plate. The chromosomes. This is where shows the two duplicated chromosomes here at the metaphase plate. So they are attached to the kinetochore. So this, chromosomes are attached to the kinetochore. They are not attached to the kinetochore microtubules radiating from the centrosomes at the poles. Radiating from the centrosomes at the poles. So this kinetochore, they're attached to the chromosomes radiating from the centrosomes at the poles of the cell. At the poles of the cell. At the poles, they came from this at the poles of the cell. So then non-kinetochore microtubules not attached to the not attached to the chromosomes. This is the a non-kinetochore non-kinetical microtubules not attached to the chromosome, overlapping at the metaphase plate. They just overlap, but they are not really, attached to the, to the chromosome, but they just overlap here. They just overlap. That's all it does. So not, so instead of connecting to the chromosome, so the non-kinetical, you just overlap at the metaphase plate. That's what it does. Yeah, basically. Here again, the second picture, which is here, the transmission electron micrographs reveals that the kinetical of the chromosomes of the two sister chromosomes face opposite poles of the cell. Opposite poles of the cell. So this this kinetical, they just face at the opposite poles of the cell. That's what it does. Again. So in anaphase, sister chromatids separate and moving along the kinetical microtubules towards opposite ends of the cell. So the microtubules shortens by the depolymerizing at the kinetical ends. So non-kinetical microtubules from opposite poles overlaps, as we say it again, pushes against each other, elongating the cell. So in telophase, so the reason why they overlap is just to elongate the cell. So in telophase, the genetically identical daughter cell's nucleus forms at the opposite ends of the cell. So cytokinesis, let's look cytokinesis, a closer look. In animal cells, cytokinesis, division of cytoplasm, typically follows mitosis. So in animal cells, cytokinesis occurs by a process called cleavage. So the first sign of cleavage is the appearance of cleavage furrow in the cell surface near the old metaphase plate. At the cytoplasmic site of the cleavage furrow is contractile ring of actin microfilament associated with molecules of the motor protein myosin. So contracting the ring pinches the cell into two. The cytokinesis in the plants. So cytokinesis in plants which have cell walls involves a completely different mechanism from animals. Cytokinesis. During telophase, vesicle from Golgi apparatus, Golgi coalesce at the metaphase plate, forming a cell plate. So the plate enlarges until the membrane fuses with the plasma membrane at the perimeter, and the content of the vesicle form new cell wall material between the daughter cells. That's how basically it is. This is again the micrograph shows the micrograph shows the cleavage furrow, I'm sorry, I just can't see the point, that's why I keep doing that. So the cleavage furrow of divided animal cells, as it appears on the cell surface just inside the plasma membrane at the location of a furrowing, the microfilament forms a ring. So this actin filament interacts with the molecules of myosin, causing the ring to contract. So the cleavage furrow deepens until the cell is pinched into two. Here again we have, in this micrograph again, we have, cell plate, which is plant cell. So we have in micrograph the soybeans root of the telophase. You can see now the nuclei at the two poles, and a vesicle from the Golgi apparatus that are coming together to form a cell plate at the equatorial plane of the cell. So this vesicle ladens with the cell wall material